Hi everyone, this is Nehad from Bio with Nehad channel. In this video, we're going to continue what we started in the biochemistry, the 4 biological macromolecules. We have been talking about the carbohydrates, then the lipids, then the nucleic acid in the last 3 videos. Today, we're going to explain proteins. We said they are macromolecules because they are so big molecules, and we said they are polymers because they are made up of, as you see here, monomers, similar cells. They are connected together. So the proteins here, they are made of amino acid. So we'll be explaining the structure of the amino acid, which is the monomer of the proteins. We will be going through some examples of proteins. Let's get started. So our polymer today is the protein is made of a monomer. What's the meaning of monomer? It's the repetitive similar unit that it's connected together or bonded together to give the polymer, which is the protein. Okay, so our polymer is protein. It is made of a monomer called amino acid. Basically, this is the structure of amino acid that is made of an alpha carbon in the center of the molecule of the amino acid. And as we discussed before, it is it is surrounded by 4 valence electrons, so it has to have 4 covalent bonds. The first covalent bond is connected to hydrogen bond, a hydrogen atom. The second covalent is connected to an amino group, then a carboxyl group, and the last one is the R group, or we can say it's the functional group. What is the function of the functional group? So if we have, and actually this is real, that we have 20 amino acids. So the 20 amino acids, they have to be different because we said they are 20. So they are 20 different amino acids. If we have the same 4 components, so it will be the same structure. We can't have the 20 structures. So basically we have one different structure, one different group, which is the R group, that made the 20 amino acids different from each other. So the R group, we have here 4 different choices. We are changing, okay, to have the 20 amino acids, and those are the 20 amino acids. So I'll just change the color to be clear. So I have the amino, the carboxyl, and the hydrogen. So the first one, the R group is H. Then I have those in common, and the R group, so the R group is CH4. I have again those in common, and I have, this is the CH3. This is the R group. Then those, I have them in common. Then this is the R group, CH2, CH3, and so on. So that's how I have 20 amino acids. The most simple one is the glycine one. The R is only an H. Then the R is different from one to another. And because it has an amino group and a carboxyl group, it is actually either hydrophobic or hydrophilic. When the amino acids are connected together, the carboxyl group will be connected to the next amino group of the next amino acid, okay, by dehydration. Actually, the bond is called a peptide bond, and the chain that will be formed is the polypeptide chain. So those bonds, those are actually the amino acids. The cartoon faces are the amino acids, and those are peptide bonds, and the whole thing is the peptide chain. Regarding the function of proteins, we have different actually multiple functions. If you remember, the ribosome is the organelle in all your cells that it's like the site of protein synthesis, right? So you will be having your proteins from translating the DNA sequence into proteins in the ribosomes. This protein can be a pigment, your eye pigment, for example, the brown color, can be what else, can be your muscles, can be an enzyme, can be a hormone, because like most of the hormones are made of proteins. So many structures in your body, it's made of protein. We have the structure of protein. It's actually 4 levels. The first level is primary protein structure, which is only the sequence of the amino acids connected together, so it's one dimensional. Then the secondary protein structure, it's 2-dimensional, and it's, as you see, a spring-like shape, because some parts are repelling and some are attracting each other. Because if you remember, we have the amino groups, we have the positive ones, we have the carboxyl groups, negative ones. Then just it will be folded and looped to give its function. Okay, and this is the tertiary protein structure. It's 3-dimensional. Many of these 3-dimensional, actually, to be proper to its function, it's the quaternary group protein structure. Okay, so it will be folded and coiled more and more. The tertiary, or like the strands and with the folds and loops, will be folded again and coiled again. Here we have more than one, just to meet the function. Okay, so it may be joined to other proteins, or added to carbohydrates, added to vitamins, added to minerals, all of this to meet its function. Thank you for watching. Please subscribe, follow, and like all my videos, and wait for the next video. Enzymes.